Hello everyone! I am one of the 12 PhD students that work within a group of 12 international students to improve the way we treat breast cancer today. Our project, called EpiPredict, it's on its mission to understand why current methods fail to treat the patients. The ultimate goal is to provide better and personalized methods of treatment to the patients that have breast cancer. Many of you may have a family member, a friend, or someone that you know that have dealt with breast cancer before. This condition has been around for a while and more patients are diagnosed every year. Actually, in women, it's the most common form of cancer worldwide and the second leading cause of death. 15% of women will be diagnosed with breast cancer during their lifetime. In the UK alone, 50,000 women begin annually breast cancer treatment based on endocrine therapy. At the time of diagnosis, the tumor of each patient is molecularly profiled. More than 70% of the cases are typified by the expression of a receptor that is responsible for binding a hormone called estradiol. And this is the primary stimulus for breast cancer cells proliferation. So after the removal of the primary tumor, these patients are treated with endocrine therapy. Endocrine therapy involves the administration of drugs that will interfere with the action of the estradiol. The most commonly used drug is tamoxifen. So, tamoxifen binds to the receptor. This inhibits the hormone effect by blocking the estrogen-induced tumor growth. The tamoxifen-based therapy is by far the most effective approach to treat ER-positive breast cancer. Actually, 85% of the patients survive the disease for 5 years or more. We are still far from providing an absolute cure for the disease, but we're pretty close to extend the survival of the patients up to old age. The main issue with the current therapy is the rising of a condition called drug resistance. This resistance allows the cancer to come back in a much more aggressive way and it's no longer affected by the drugs. In other words, incurable as of today. Cancer cells have a completely different behavior from normal cells. They have a lot of flexibility and they create a special environment that allows them to survive. So under treatment, the cancer cells will develop new strategies that will allow them to bypass the therapy. And this makes it extremely hard to be defeated. However, how the cancer cells become drug resistant, it's still mostly unknown. Part of this resistance might be explained by the composition of the genetic code, but this does not give us the full picture. But before I can continue, we need to understand how DNA is organized within the cell. So in human cells, DNA is packed around these spools that we call histones, and they will be responsible for packing the DNA inside of the nucleus. These DNA structures will determine which genes are going to be active or inactive. But to understand this better, we have to go deeper and talk about another field, epigenetics. The term epigenetics refers to all of the chemical modifications of the genome that are inherited when the cell divides. These modifications do not change the DNA code, but they do change how the DNA folds. And the new structure means different active and inactive genes. Recently, scientists discovered that there are epigenetic changes within the cancer cells during treatment. For example, methylation and acetylation of the DNA in histones. This makes cancer to behave in a different and sometimes more aggressive way. We in EpiPredict aim to understand the epigenetic changes of breast cancer cells that cause therapy resistance. With this knowledge and by combining different disciplines, we hope to be able to predict the outcome of medical treatment in breast cancer. Based on epigenetic traits, we want to be able to both monitor and predict resistance to endocrine therapy. We also want to build and use new epigenetic maps to develop supplementary therapy plans. To do this, the 12 early-stage researchers in the EpiPredict consortium will provide new solutions to this urgent problem. We hope to achieve this by applying state-of-the-art research such as genome engineering, computational biology, and epigenetic profiling. We also have to make sure that our results will eventually benefit the patients, and this is not only a huge scientific challenge, but also an ethical and social one. You see, in our research, we are dependent on tissue samples and also clinical information from patients that is not always readily available. We also need to make sure that we have the consent from the right people in order to use them. But we have another problem. We wonder what's the distance between our scientific activities and the clinic, and when should we start working together in order to make our work relevant to the medical field? Well, that's the reason why we organized this Science Cafe. We hope to bring the different disciplines together and debate the current state of both breast cancer research and the clinic. And with that said, we want to formally welcome you all to this Science Cafe. We hope you enjoyed the discussion, and for now, EpiPredict out.